I may have. Yeah.
All right, well, why don't we get started today? Welcome, everybody. I'm glad to see so many faces here. I know this is a different time for us, but I'm glad that people read the highlighted part of the messages I bombard you. I'm going to introduce Joy Pate to everybody, and many of you know her or met her these last couple of days, but a couple of you that have not. She's here for two big reasons. One is she's a world-renowned ovarian physiologist and a reproductive immunologist. And so I've been thinking about trying to get her here for years. But the second big reason is she announced her retirement a couple months ago, and it scared the crap out of me. I'm like, oh, we got to get her here before she actually eases into retirement and doesn't want to do these kind of things anymore. So it's really perfect timing to have her come in this semester. So thank you, Joy, for doing that. Tell you a little bit about her, and I got my notes so I don't screw up too much. She did her bachelor's at the University of Delaware, and went on to where I think you started a master's, then transitioned right into a PhD at New Hampshire. Sorry, I almost said Rhode Island. And after that, she directly went into a faculty position. And she told many of us yesterday that she had some really nice postdoc options lined up, but Ohio State came calling, and she answered the call and joined the Department of Dairy Science as the first female faculty member, I believe, and rose through the ranks there. And actually, even for a time, tried out administration as an assistant department head. Does that sound right? Oh, yeah. We won't talk anymore about that. But in 2008, I think it was, she was lured away to Penn State. And she, where she received a chair position, the Rumberger Chair in Agricultural Sciences, and also became the director of the reproductive biology and health incentive. There's actually a group of 5 or 6 scientists that followed her shortly thereafter, and many of them are still there. And so they had Joy come in to actually get a really nice reproductive program developed at Penn State. And so what we asked her to do for this seminar was a little bit different. You know, she could do like in her sleep, a regular seminar on her work over the years and decades. But what we were more interested in have her do is give a career seminar, what she's learned from her work, along with telling us about what she does and why it's important. I was kind of hoping that she would also include things about what she's learned along the way. And not to put pressure on you about this, but she has said that it's been a challenging seminar for her to put together because, you know, it's so different from the others. But she's up for the task. Those of you who know Dr. Pate know that she's going to be do a great job with this. So with this, I'll let you get back up here, Joy, and take over.
All right. Thank you. And thank you, Alan, for inviting me. And as I recall, our conversation on the phone about this seminar was, well, there's no one there who studies the corpus luteum really, and there's some repro people, but there might be a lot of non-repro people in the room. So if I stood up here and showed you data about the corpus luteum for an hour, you were all going to fall asleep. I realized that. So Alan and I came up with this thing, well, but what about, you know, some tidbits that I've learned over my career? And last week, I sat down and thought, what did I learn over my career? So I've never done anything like this before. We're going to see how this goes. Okay. I am going to tell you about the corpus luteum, but I'll try to tell you some other things as well. So here's what I'll do. And I need to walk out here because I don't like being up behind the podium. So let me know if you can't hear me or anything. We'll talk a little bit about what is the corpus luteum, because I know some of you were not familiar. And then I'm going to try to give you an overview of some of the main things that I found out over the years when I was studying the corpus luteum, mostly with like schematic illustrations and not bar graphs and tables and things like that. Okay. And then what I've been working on the last few years is the corpus luteum of pregnancy and how is it that the rescue of the corpus luteum is achieved. And as part of doing that work, I ended up one day at a scientific meeting in this very, very heated scientific debate with a colleague. And I've told this story now a couple of times when I've given talks. And I think that's what got me sort of also thinking about what we learned, because the scientific debate is still ongoing. But I thought about having this debate with this colleague, having this controversy, what it drove me to do. So it drove me into doing things I never thought I would do. And it drove him to do things as well. And that's still ongoing and what I've learned from that. And that made me think that probably all through my career in doing science, I was learning about cells, but I was learning other things as well. So that's what I've tried to do today. Let's see if I can do that. Okay, so for those of you who are not familiar, the corpus luteum is a structure in the ovary, and this is a schematic cross-section of an ovary. So you probably know that the ovary is where the eggs are located. So the oocytes, or we call them, grow in little cohorts, and they go from primary follicles to secondary, tertiary follicles. And then these large tertiary follicles are what ovulate and release the oocyte. And then there are cells that line this follicle that stay there and go through this differentiative process to form the corpus luteum. So that's what the corpus luteum is. Let's see if I can go. I study the corpus luteum in cows for a number of reasons. It's very important, but also this is cross-section of bovine ovaries. So this is an ovary without a corpus luteum, and this is an ovary with a fully functional corpus luteum, the estrous cycle. And one thing you might know is the corpus luteum itself takes up it's about the size of a half of an ovary. So you get tons and tons of cells to work with. So it's a really great model. And this corpus luteum forms after every ovulation. So with every cycle, it goes from a few cells in a follicle that you can't see with your naked eye to this tissue that's anywhere from like 5 to 10 grams in a few days. And then at the end of the estrous cycle, this will regress. And again, in just a few days, it regresses to, here's the one, the CL from the previous cycle. So it's incredibly dynamic structure. And what the corpus luteum does is synthesize progesterone. And this is important because progesterone is necessary to establish pregnancy and maintain pregnancy in all mammals. So everybody and every animal who ever wants to conceive and be pregnant needs a robust and really great corpus luteum. Now, I mentioned the cyclic nature of the ovary. So this is just depicting what I just told you happens, that follicles grow, they release an oocyte at ovulation, and then the corpus luteum forms. And during that time, you go from a period of high estrogen production, because the follicles produce estrogen, to a period of high progesterone production. And this corpus luteum sits there for a while. It's maintained, it's making progesterone, but at the end of the estrous cycle, or at the end of a menstrual cycle in primates, that corpus luteum regresses. And so progesterone declines very rapidly. In ruminants, which I work with cows, that death of the corpus luteum is brought about by the uterine release of a hormone called prostaglandin F2 alpha. That causes the CL to die. Well, if there's an embryo, if this oocyte gets fertilized and that embryo is present, we need that corpus luteum to not die, or the embryo will also die. So they have a symbiotic relationship. And so that's depicted what happens here, that if there's an embryo present and the embryo signals, that corpus luteum doesn't die, instead it's maintained, it's rescued by the embryo, and progesterone is maintained. And so we will come back to that. Okay, so some of the really cool things about the corpus luteum is that it forms and it grows, as I said, remarkably fast, and it exceeds the rates of growth of every known tumor. So it's actually, it could be a really, really great model for tumor development. It's a model for angiogenesis and hyperplasia because it's very highly vascularized, and that happens in a matter of a few days. It's a model for differentiation of cells. Because it is steroidogenically active and very metabolically active, it makes tons of free radicals. And it's a model for how to control those free radicals and keep them under wraps. So it does all kinds of good things. At the same time, it regresses equally as fast. So when it's time for luteal regression to happen, progesterone drops very, very rapidly. And then all of those cells start to die, and all of that's cleared again very, very rapidly. So, and when that happens, none of the surrounding parenchyma or cells in that ovary are affected by this massive, what we now know to be an immune response that occurs to get that CL to regress. So again, good model of tumors. Okay, in cows and in sheep, the corpus luteum on the ovary has this very interesting relationship with the uterus. So this is one uterine horn being depicted here. And that's because the uterus is what secretes prostaglandin F2 alpha, which will kill the CL at the end of the cycle if there's not an embryo present. And the prostaglandin F2 alpha is secreted out of the uterine vein. And then there's this relationship where the artery that's going to take blood up to the ovary wraps around this uterine vein. And that allows for the prostaglandin to be directly transferred from the uterine vein into the ovarian artery and then go up to the CL. Now, that's important, and it's going to be really important when we get into this scientific debate, because when prostaglandin F2 alpha travels through the lungs, it gets metabolized rapidly to what we call prostaglandin F metabolite. It has a longer name, and that metabolite is not active. So if the prostaglandin is released from the uterus and goes through the lungs and then had to go back, circulate back, there probably would not be enough prostaglandin to kill the CL. In sheep, that's absolutely true, because in sheep, 97% of that PGF2 alpha gets metabolized in the lungs the first time it goes through. In cows, it's about 50 to 60%, so a little more escapes. But that's going to become very important to our story later, okay? So let's see. Okay, let me go back. So now I'm going to go back to what seems like childhood days when I was in grad school. And when I went to grad school, I had no idea what the corpus luteum was. I'd taken a repro class and I promptly forgot anything about that. But I knew I wanted reproduction. So I went to grad school, and it turns out, you know, I'm going to study the corpus luteum, and 40 years later, I'm still studying the corpus luteum. So I'm a very slow learner, apparently. But I was intrigued by, we knew prostaglandin that came from the uterus killed the corpus luteum. We didn't know how it did it. We didn't know how prostaglandin acted on the corpus luteum. And so that's what I decided I wanted to study for my PhD. So what the tools we had at that time were, here's this ovary of the cow. So you could do something to the cow. You could inject the cow with prostaglandin F2 alpha and the corpus luteum will regress. And so you could study some things about that, but that doesn't really tell you what's happening on the cells. You could take out the CL and you can dissociate it enzymatically into single cells. So these are luteal cells that aren't they the most beautiful cells you've ever seen in your life. So they come in different sizes. We call these, they have very original names. This is a large luteal cell, and this is a small luteal cell. And large luteal cells in the cow can be as big as 70 microns. So they're really, really huge cells. So at that time, people had dissociated these cells and they would put them in tubes and do short-term incubations with them. But when they added prostaglandin F2 alpha, you would expect progesterone production to go down and maybe the cells to die, right? Because it's the luteal litigation. But in fact, it stimulated progesterone production in these short-term systems. So I thought maybe it was that prostaglandin F2 alpha needed a longer time to act. So what I would have to do is take these cells and put them in culture and culture them as monolayers so that I could study prostaglandin action. But nobody had ever done this with luteal cells at the time. So I was taking the cell culture class my first semester in grad school, and that's what hooked me on science, because I looked through this microscope and we did little cardiac muscle cells. When you learn cell culture, it's a great one, because as soon as they become confluent, they start to beat. And I saw these cells beating in there, and that was it. From that day forward, I was going to be a scientist. I was going to study cells. So if I grew those, I ought to be able to grow luteal cells. So I associated them, and I learned what I learned in cell culture class, and I put them in culture, and they were beautiful. So here's a large luteal cell here. Its nucleus full of lipid droplets, because it needs a lot of cholesterol to make progesterone, and small luteal cells all around. And so there they were. They were alive. They made progesterone. But the corpus luteum makes progesterone in response to a pituitary hormone, LH, that we call luteinizing hormone. And when I added LH to these cultures, it did not increase progesterone. And so it's kind of hard to study what prostaglandin does to inhibit progesterone if you can't even get the thing that you know stimulates progesterone to do its thing in culture. But I'm rather stubborn and I didn't give up. And to you grad students who have techniques that aren't maybe working right now, I want to tell you, it took me 3 years to get LH to stimulate progesterone in these cultures. And it was a simple fix. I just removed the serum and then everything worked. But 3 years, I kept trying all different things to get this known luteotropin to stimulate progesterone. But during that time, I couldn't study prostaglandin action right away, but I could study other things about these cells. And I just mentioned that the cells need a lot of cholesterol because that's the precursor to make steroid hormones like progesterone, and it makes tons of progesterone. So although they weren't responding to LH at the time, one of the things I got interested in were lipoproteins. And everybody's heard of these things now, LDL and HDL. Back then, I had never heard of them, but there was another lab in my building where they were studying lipoproteins and lipoprotein action. So I thought, well, if lipoproteins carry cholesterol around in our blood, you read about these like the bad lipoproteins, good lipoproteins, then maybe they carry cholesterol to these cells so that they can make progesterone. So I started culturing these cells with lipoproteins, and along this way, we discovered that yes, luteal cells do use LDL and HDL as a source of cholesterol. It's the predominant source. It can use both of them. So the CL is absolutely dependent on these lipoproteins. So I was able to find some stuff out. Then I got LH to work, and then I could study PGF2 alpha. And we were able to find out using these cultures that PGF2 alpha doesn't inhibit any of these processes, but it inhibits this cholesterol from getting through the mitochondrial membranes. That's what it does. Okay, so that was like my whole PhD thesis there. But here's what I learned from that. And so here's the first of our learning lessons. First of all, don't give up easily. So as you're going to see, I'm going to be talking mostly to the students here today, okay? Research is hard and experiments don't always work. And you can't walk into the lab and follow a procedure or a protocol and assume that it worked. So you have to validate that. And you're going to find out sometimes it doesn't do what you think. So don't give up, but learn from your failures, because we learn better from our failures than we learn from successes. We don't remember those as well. And figure out how to make techniques work. I mean, that's what you do as a scientist. You don't just pick up a kit, you figure out how to do something to answer the questions that you have. Maybe 3 years was a little long, but we got something out of it. And the other thing is, you're going to hear this one again, learn about the research outside of your own area. Because if I had not done that, I wouldn't have known anything about lipoproteins. And yet I want to be humble here, but I became the person who discovered that luteal cells of any species use lipoproteins as their source of cholesterol, because there was this lab down there and I learned about lipoproteins. So learn outside of your area. Okay, so I graduated and I went to the Ohio State University. And the second life lesson that I learned in grad school that I quickly applied here was, so I don't know if this was a time like back in those days or if I was just really stupid, but when I got offered this faculty position, there was no such thing as a startup package. They didn't have those then. So I got a faculty position and I got a room with some shelves in it and some sinks, and that was my lab, and there was nothing in it. There was not a graduated cylinder, there was not a pipette, there was nothing, and no money given to me to get those things. So I was scrounging around to get stuff from other labs. But one of the things I did is I went to the department chair and I started asking about how hedge funds were distributed in this department. And he looked at me and he couldn't believe it. And he said, how do you even understand that? Most of my faculty don't know about this. But colleges of agriculture get what we call formula funds. I'm not going to go into all of that. But I had talked to my advisor a lot about how funding came in the lab and how these things work. And I was able to go in and say, but I know you get this certain amount of formula funding, and how are you using it? And can I access any of that to start equipping my lab? And the answer was yes, I could. All I had to do was ask, and he wasn't going to just offer it. So, one of the things I learned early on, you need to get out of your own little box of your own cells and your own embryos and whatever you're doing. And you need to talk to your PIs, to your mentors and your advisors and faculty. Learn the ropes, learn how departments function. They all function a little bit differently, but there's a lot of commonalities. So when you get a position, whatever kind of position that is, you have some of that background and you go in with some knowledge and you can negotiate. So I learned that. The other thing is when I think about my first years as an assistant professor, my early years, I always think of Nate Fetchheimer. And so I'm going to tell you a few things about Nate Fetchheimer. So I told some of the people I had dinner with last night, Nate Fetchheimer was the most eccentric, unusual, and intimidating individual I had ever met in my life. And I am not an easily intimidated person. But he was this little guy, he was a chain smoker. You could hear him coming down the hall. So this was before they banned smoking from buildings. Or it probably was banned, but nobody confronted me. So, and you could hear the click of his lighter. And he had these routines. And I knew exactly like what time of day he would come walking up the steps and you'd hear the click. And then he'd go in his office and shut the door and it would fill with smoke because he smoked all day. And so everyone was afraid of Nate. Everyone also revered Nate as a scientist. And I was in a department that was very applied research, and I'm really a cell biologist. And Nate was the one other guy who kind of was a cytogeneticist and who kind of I probably could have related to, but the guy was just so scary and weird, I wasn't going to relate. So anyway, one day, as I was scrounging for equipment, I found in the basement of this really old building I was in, there was a storeroom, and in the storeroom, there's equipment and stuff there. And there was this piece of equipment that I needed. So I went to the department chair and I said, do you know who's, I don't even remember what the equipment was, but say the balance, do you know who that belongs to? Because I really need a balance. And he says, oh, if it's in the storeroom, it's fair game, you can use it. And I said, okay, great. So I went and got the balance and I put it in my lab. And I'm in my lab with my students and we used this balance for probably months, I don't know. And one day, my lab door opens and in walks Nate Fetchheimer, lit cigarette hanging out of his mouth. He walks by, I'm in there with a couple of undergrad students talking at the lab bench, and he walks over and he picks up the piece of equipment and he turns around and walks out. Well, that's interesting. And so I told my students, I'll be right back. So I followed him and he went into the main office where the secretaries sit and the department head is in the room. And he was doing something with this piece of equipment in his hand. I said, excuse me, why did you just take this piece of equipment out of the lab? And he said, well, it's my piece of equipment, it's not yours. What do you think you were doing? Just like taking this piece of equipment without ever asking. And I said, well, I did ask. I asked the department chair and he didn't know who it belonged to and he said I could use it. And I said, it's really rude to just come in and take it and not say anything to me. I'm really sorry that I did not know it was yours. But by the way, don't you ever walk in my lab with a cigarette in your mouth again, because that's against the rules. You don't have to do that. And I went home and I thought, that's it. I will never get tenure. and I told them last night, the next morning I came in and there was a sealed envelope in my mailbox. And inside that envelope was a 4-page handwritten note from Nate Fetchheimer apologizing to me that he hadn't talked to me about this piece of equipment. And so Nate and I actually became really good friends after that, and he became a very important mentor. 2 other little quick ones one time. So I kept watching, as Alan said, I was the first female in the department. I was the second female in the entire College of Agriculture. I was 26 years old. I had braces on my teeth and I was single. So I did not fit in with any of this. The next youngest faculty member in the department was 55 years old. So I was like their granddaughter. They didn't know what to do with me. And I kept seeing them go into this conference room every day at 10 o'clock, like a bunch of faculty. And so finally I went to the department chair and I said, what is it that everybody does every day at 10 o'clock? Oh, well, we go and have coffee and talk. I said, oh, my advisor did that when I was in grad school. He went down and all the big problems got solved in coffee hour. I knew that. And I was being left out of that. So problems get solved at coffee hour and in men's rooms, before this time. So I said to the department chair, well, am I invited? Can I come to coffee? And he said, well, sure, you can come. So the next day I got my coffee and I walked in and sat down. And this is how then I got to know Nate and stopped being intimidated by him, as well as the other faculty. And one day I walked in a couple of years later and I was a little bit late. And they looked at me and they said, Joy, we voted and we decided that you're now one of the boys. You're one of the boys. And I said, well, thank you for that. And Nate, being very wise, looked at me and he said, you can be one of us without being a boy. So I thought that was really nice. The other thing he told me was, when you live in Columbus, you eventually have to learn to play golf. So I learned to play golf and I got really kind of hooked on this. I was lousy at it, but it was pretty good. I enjoyed it. And so I told them one day in coffee, I learned to play golf and I'm out there on the Scarlet and Gray course. And after my story, Nate looked at me and said, and someday you may need to choose because, golf takes a lot of time. Do you want to play golf or do you want to get tenure? And I won't get tenure. So the lessons from that, two of the things I learned from Nate. What it means to be a university professor. You're not just there doing your research and teaching your students, you are a university professor. And that means service to the university, service to your department and to your profession. And we talked often in long conversations about the ethical obligations that you have when you become a scientist. And so it's a very important lesson I learned from Nate. And also, if you're going to do science, to take the time to be precise, be thorough, make your work be the very, very best it can be. Validate every single protocol you do with your own hands. Don't just pick up a protocol and do it and think that the data are any good. Always validate that. Recognize that sometimes you can't be perfect and that's okay, and be able to move forward. So the lesson I think to give you is to choose your mentors very wisely. So he was one of mine. Okay. So now I'm there and I'm culturing these cells and I want to study prostaglandin F2 alpha action and I can put PGF2 alpha on the cells. And if PGF2 alpha is the luteolysine that causes luteal regression, what do you think is going to happen to the cells when I put it on in culture? Mary Alley?
They will die.
They should die, right? But they don't. They never die when you put PGF2 alpha on there. So PGF2 alpha does not kill the cells. And that always intrigued me, why they didn't do that. So we could study its action, but it never killed the cells. So I'm going to go back to this other previous life lesson. I went to a seminar and the guy was talking about MHC molecules, major histocompatibility molecules. I had never heard of these before because when I was in grad school, my advisor told me to take a course in immunology and I very craftily talked him out of making me take an immunology course. And that was a big mistake. So this guy's up there talking about NHC molecules, and you probably know they're the molecules our cells express that cause us to know self from non-self. So it's how our immune cells recognize cells. So I listened to this, and then I got in my car, and I'll never forget, I was driving to the airport to pick up Joe Ottebrand, who was coming to interview for a faculty position. And on the way to the airport, I thought, maybe when the corpus luteum regresses, it starts to express these MHC molecules and it becomes considered non-self. And then the immune system attacks it and gets rid of it. Sounded great, right? Like maybe that's what happens. Very naive, but I got this whole idea in my head that luteal cells could be antigen presenting cells, that they could express class 2 MHC molecules, and they could activate immune cells like that, and that's what would happen with luteal regression. So I decided that, well, my department chair said, you should write a grant proposal at some point. So I had to figure out how to do that. So I wrote a USDA grant, and the grant was all about luteal cells presenting MHC molecules, and that's how luteal regression occurred. And the reviews came back from the reviewers, and the last line of the panel summary was, this person needs to take an immunology class. So I did not get funded on that first grant. And basically what they said is that MHC molecules are either there or they're not. So the dogma is that B cells and macrophages, and many of you know this, express class 2 MHC molecules, but the rest of our cells don't. And they don't disappear, they don't change, and if you would go take an immunology class, you would know better. So I went and sat in on an immunology class, found an immunologist at Ohio State to talk to. And the other thing I did was I got my graduate student to say, let's see if we can find these molecules on those tissues. About the same time, immunologists were learning that many autoimmune diseases occur because class 2 molecules start to get expressed on cells that didn't otherwise express them. And so we looked for those on cells in the corpus luteum. And this is how many of those cells were positive for class 2 MHC. And if you remember that estrous cycle, this is in the developing CL, a fully functional CL, and a CL just before it's going to die. And these are different populations of cells, don't worry what they are. But the bottom line is you can see as the CL develops, it has very little to no expression of class 2 MHC. But as the CL grows, some cells start to express it. And as the CL is about to regress, there's lots of class 2 MHC on these cells. And what was even more interesting is if this is again, these cell populations here, we compared the day 18 CL in cows that were not pregnant. So the CL is about to regress. The cows that are pregnant where that CL will be rescued. And in that case, the NHC molecules were less. So that told us that NHC may actually have to do with luteal regression, and it may have to do with rescuing the CL. And so we rewrote the grant and we got it. And so the lesson from that was, again, learn the research outside of your area, because I went to this seminar, had never heard of MHC in my life, ended up writing a grant, ended up discovering that this was here. And this actually launched like the next 30 years of my career into immunology. So I kind of became an immunologist along the way. And use your intuition, listen to your gut if you get these ideas, but make sure you have the knowledge and get some preliminary data if you can to support those hypotheses. Okay, so I told you it launched me into immunology, so I'm just going to quickly tell you that this led us to start thinking about what immune cells produce and what they might do to luteal cells. And we found out that in fact, the proteins that immune cells produce inhibit progesterone production, and they increase or they stimulate prostaglandin production within the CL, within luteal cells. So things you would expect to happen if they're involved in the regression of the CL. And better yet, remember PGF2 alpha did not kill the cells. When you culture the cells with these cytokines, they died. We were so happy our cells died. So these cytokines that come from immune cells are, we now know what are really causing these cells to die. We also wondered if the immune cells that got into the CL, so these are a bunch of immune cells inside a CL, are they just sort of all the immune cells that are like floating through in the blood? So are they the same as the ones in the blood? So these are blood immune cells, just proportions of T cells here, CD4, CD8, gamma delta cells. If you don't know immune cells, it doesn't matter here. But what you can see is that the proportions are a little different of the cells that are within the CL versus the cells that are in the blood. So that means the CL is selecting, we used to say that the CL selects which cells can take up residence in its neighborhood. So which cells we're going to let come and live in there. And so that led us to think about this whole thing, that in a fully functional CL, the cells that come in and that take up residence inside the tissue that leave the blood would be cells that are involved in homeostasis, in maintaining the corpus luteum. So immune cells are not just for killing things. But when the CL gets ready to regress, there's different immune cells in there. And so if this was true, then does it mean that immune cells come in and then prior to regression, they leave and different immune cells come in? Or could the CL actually direct the function of those cells living in its neighborhood? And so that was our hypothesis. So it selects against these the pro-inflammatory cells, and then the cells that maintain homeostasis decline. Okay, but this question, can the luteal cells themselves direct the function of lymphocytes? And nobody had looked at it this way. And it turns out that if you culture those together, so you take lymphocytes from the peripheral blood of cow and you put them in culture with luteal cells, these are T cells by themselves, and then T cells after they've been exposed to luteal cells. And one of the things you can see is they get big. And that means they blasted, that means they're proliferating, that means they're activated. So it turned out that luteal cells do in fact activate T cells. And if the luteal cells that you put in culture came from a functional CL, it changes the phenotype of those lymphocytes so that they produce cytokines that are that would tend to suppress a pro-inflammatory response. And it decreases cytokines that would cause a pro-inflammatory response. On the other hand, if you get the cells from a regressing corpus luteum, you get different effects. The cells change more like you would have a pro-inflammatory response. So what we found out was that luteal cells are very potent stimulators of T cell proliferation. They change the phenotype of these cells, and the resultant phenotype is dependent on the functional status of the CL. So the CL is in control, and those luteal cells are programming the T cells. Okay, now I'm going to go back. So that was finding out how luteal regression occurred. But my real question was, we want to maintain a pregnancy in cows. Fertility in cows is not great. We need to do everything we can to maintain pregnancy. How is that CL maintained? And remember that PGF2 alpha comes from the uterus. And to remind all of you students out there of these classic experiments, we know from Princess Milo Levine's father in 1956, that if you hysterectomize a cow, if you remove the ovary or the uterus from a cow or sheep, but you leave the ovaries intact, then the CL does not regress for a while, right? You all know that. You will all learn that in the repro classes, which tells us that uterus is absolutely necessary to get the CL to regress, and it's because it's the source of PGF2 alpha. Now keep that in mind, because we're going to come back to that. So we also know that in pregnancy, the blastocyst produces interferon tau that acts on the uterus. And basically what it's doing is altering prostaglandin release from the uterus. And note that I said alter. And you all teach that it inhibits prostaglandin, don't you, Dr. Gillen? Good. But it alters prostaglandin production. And there's a question, could that interferon tau also act on the ovary itself, on the corpus luteum itself? And it probably can, because if you culture cells with luteal cells with interferon tau, you get a lot of changes that would be indicative of things that would have to happen in order to rescue the corpus luteum. Just keep going from there. So I'm going to take one little aside and then come back to this final story for you. And this aside is, while all this was happening and I was doing all this immune cell stuff and starting to think about luteal rescue, I started to have some self-doubt. So I'm not really an immunologist, and I was doing all this immunology, and here I am publishing these papers that these steroid producing cells are programming immune cells, which is kind of blasphemy. And I started thinking, you know, I really need something else to work on, because this immunology thing could be all wrong. And what if I get to the end of my career and I was wrong about everything I've been done? So I need something else. And I had this in the back of my mind, what I would get for something, do for something else. And I was walking one day with my postdoc from getting ice cream, and he had been to a seminar that was some plant science seminar. And he started saying he learned about MicroRNA. What are that? What's that? What's MicroRNA? And he told me about them. And I thought, wow, that could be a key. I mean, MicroRNA just sounded really cool. And I'll tell you what they are in a minute, but basically I came up with this idea that there's two really important times in the life of a CL. One is when it goes from this developing CL that won't respond to prostaglandin to a fully functional CL that does. and another when the CL is going to live or die. And it's a pretty critical time in your life. You're going to live or die. And what I was learning about microRNA is that they might regulate that. So the students, I'm sure, have had all this, but microRNA are basically small sequences of RNA that hybridize to mRNA targets, and they can cause those RNA to be degraded, but more likely they just block them from being translated into proteins. So it's a post-transcriptional way of regulating protein production. And it can happen very, very quickly. So I thought that fits with these critical control points. And I'm not going to show you any of those data, but I just wanted to tell you by, kind of saying I needed to do something different for my research. We got into microRNA and wow, what's fun. I still have like 4 or 5 more papers I have to write about microRNA. It's the coolest stuff in the world. But that's what led me to these life lessons I'm supposed to be telling you about, which is always have at least 2 irons in the fire, because one may not pan out, or maybe it was wrong. I think I was right, but it's good to have 2 irons in the fire. Once again, think outside of your discipline. Go to seminars, talk to people who are doing things you don't know about, and always question your own interpretations and directions of your data. It's so easy to get on this line of thinking and you get data that support your hypothesis and you think you're right, and you're right, and you start to get blinders on as you go through. And it's really important to question those things. And it led me into this whole thing. Okay, so here we were studying the CL, and I wanted to know if microRNA were involved in rescue of the corpus luteum. I'm not going to show you those data just yet, but the answer was it looks like they are. But at the same time, what we did is we collected CL from day 17 of the estrous cycle of cyclic cows. So again, that's a CL that's about to die, and pregnant cows, and that's a CL that's about to be rescued. And we did deep sequencing for microRNA. That was what I was most interested in. But as long as we were doing it, we did mRNA-seq as well. And along came a student to my lab from Virginia Tech, Camilla Hughes. And these are Camilla's parents sitting here in the front row, if you don't know. And Camilla came into my lab to be a grad student. And I had a postdoc who was working on the microRNA sequencing data, and I had all of this RNA-seq data. I had never done sequencing before in my life, and I don't know how to do bioinformatics, but I thought, well, here's this student. So this poor first-year grad student, I handed her this RNA-seq data. I said, you have to figure out how to analyze all of these data. And she, about 9 months later, probably had many bad names for me, because I know it was a frustrating thing, but you know what? She figured it out. And she did a really great job. And so we published that the pathways that are associated with these differentially abundant mRNA in the CL of pregnancy compared to the CL of the estrous cycle in day 17 have to do with immune cell signaling, largely. And the pathways with the differentially abundant proteins, we also did proteomics, had to do with steroid hormone biosynthesis and cholesterol synthesis, okay? And then Camilla came up with this idea that why don't we put luteal cells in culture and see if the things that regulate whether the CL lives or dies regulate those genes that we found in our sequencing data to be different. And so we chose some of those, and sure enough, mRNA that we measured that were greater in the CL of pregnancy, so those would have been probably exposed to interferon tau from the embryo, were stimulated by interferon tau in vitro, but not affected by the prostaglandins. Whereas the mRNA that were less in pregnancy, so they were more in the CL that was getting ready to die, were stimulated by prostaglandin F2 alpha, but not affected by interferon tau. Okay, but one drawback of that study was that it was one point in time. So when we started to present these data, people said to us, well, the differences you see in the CL of pregnancy might not be because those things changed in that CL, but it could be because they changed in the CL that was getting ready to die. So that was a little drawback, and I'm going to come back to that because that's what led to this story. So the story time, the value of scientific debate. I'm down, but Camilla and I both were down at this meeting in Brazil. And we had already done this study that I'm going to tell you, which is kind of the last part of this. And I presented my talk and got up for breakfast. And I went and I sat down for breakfast with a colleague from Wisconsin, Milo Wilhelm. And Milo said to me, Joy, why do you call it rescue of the CL? It's not rescue, it's just maintain. I thought, why do we care what you call it? Really? But he said, all you have to do is interferon tau inhibits prostaglandin production from the uterus, shuts it off, and then the CL doesn't regress. There's nothing else that has to happen. I said, sure there is, Milo, because there's changes in the CL. We've just shown that in our day 17 CL. And we started arguing. We argued all through breakfast, all through the morning. People came to our table, people left, people joined in the argument, people left. Then it was lunchtime. We missed the whole morning session. Lunchtime came, we got lunch, we're still arguing. We argued the whole rest of the meeting about this thing, right? So Milo and I get in this argument, and what do you do when you get into this argument? You go back to your lab, like, I'm gonna prove him wrong. And he went back, I'm gonna prove her wrong. And so that argument at that meeting is what has led us into thing. And that meeting was, I don't know, 2018 or something like that. So here we go. So here's what Milo said, and he's very right. These are Milo's data. So this is progesterone production in pregnant cow. Blah, 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 progesterone goes along. This is that prostaglandin metabolite, PGFM, that they measure, and this becomes important from the jugular vein, okay? Because it's going through the lungs, so you have to measure PGFM. You can't measure PGF2 alpha from the jugular. And here's PGFM in this pregnant cow, nothing of any interest, right? Here's a cyclic cow where she has progesterone and the CL regresses, progesterone goes down, and when you measure her PGFM, you get a little blip of it here, and then you get these pulses of PGFM. And so Milo says, all you do is get rid of this PGFM and the CL is maintained. And that agreed with some older work done by Keith Betteridge, who basically said the same thing. But I also knew about work that had been done in sheep in Keith Inskeep's lab, where they cannulated the uterine vein. And so they were measuring PGF2 alpha coming right out of the uterus. And they said there was no difference in PGF2 alpha, whether that sheep was pregnant or cyclic. So they're saying the complete opposite thing. And this is, look at this is years ago, where these dates of this, and nobody had ever really resolved this. And I also knew that Keith Inskeep's lab had done a similar thing in cows, but it had not been published. It's a master's thesis that I have now, but hadn't been published. So I was curious about that thing. Okay, and keep that prostaglandin story in mind. So the final thing I was asking, and what Milo made me really think about, is the CL of pregnancy any different than the functional CL of the estrous cycle? Or does it just simply not die? So that's the question. And that's what we really got Camilla on. I think I'm gonna skip that one for time. Okay, so one of the things we know is sheep, if you give PGF2 alpha, the corpus luteum dies. But if that sheep is pregnant, and you can tell she's pregnant because see there, she's got lamb with an ear tag and everything already in it. And if that one's pregnant, in order to get that CL to regress, you need a higher concentration of PGF2 alpha. And so that tells us that the CL of pregnancy is less sensitive to PGF2 alpha than the CL of the cycle. And we also knew from data from my lab and many other labs that there are a number of other things that are different in the CL of the cycle than the CL of pregnancy. So we talked a little bit about the immune cells. CL in pregnancy make less prostaglandin. I just said they're less sensitive. There are changes in blood flow. There's increased progesterone synthesis in vitro. There's phenotypic changes in the immune cells we showed. So they are different. So here's what we decided to do. Remember that caveat we had compared day 17 CL of the cycle in pregnancy, and everybody said, well, you know, it's just one point in time. And so Camilla and I were talking about this, and we said, what we need to do is collect CL at different times during maternal recognition of pregnancy. So we can see if that CL changes over time during pregnancy, or is it the same? Is it just maintained? But I'm at Penn State where we have mostly dairy cows, and you can't get dairy cows pregnant and keep them pregnant. So I called up my buddy at West Virginia, Keith Inskeep, and I said, we want to do this experiment. And Keith had retired at this point. And he says, yes, I can help you with that. We'll get some beef cows, we'll synchronize them. We've got a really great bull, and we'll get these animals pregnant. And then Camilla and I would get up at 4 in the morning, and we would drive 3 hours down to West Virginia University Farm. We would get there by 7:30, get all of our stuff laid out. Keith would have the cows in, and he collected the CL for us on day 14, 17, 20, and 23 of pregnancy from all these cows. We did lots of driving back and forth to West Virginia. And then he maintained the pregnancies with CEDARs, progesterone releasing devices, because you take out the CL, the embryo would die. Maintained it with progesterone, and then confirmed pregnancy later on by ultrasound. And then we wanted to measure it. Messenger RNA, microRNA, and also proteomics. So we did this. And the first thing that I want to show you is that we did this principal component analysis, and these are our different CL collected. I think we have 4 or 5 per group here. Day 14, day 17, day 20, and day 23. And remember, day 17 is where we start seeing interferon tau a little bit in the blood. So out just past day 17 is where any changes induced by interferon tau in the CL might be noted. And you'll see that the day 14 CL cluster completely separately from the day 20 or 23 CL in terms of what molecules they're expressing. And the day 17 CL are interesting because 2 of them cluster, at least in this quadrant or this direction, closer to the day 14, and 2 of them clustered over here with the day 20. And that's to be expected because that's the real transitional state, we think. So we think these 2 were a little bit more advanced than these 2 maybe by some hours, but they were different. So the expression of all of their genes is different. And we asked, could it be attributed to interferon signaling? So what Camilla did was she took all of those mRNA that we had, all of the differentially abundant mRNA over time, and had them sorted into clusters. So there's programs that you can use to do this. And there ended up being 27 different clusters, which clusters are a direction of expression. So this is 2 clusters, a purple one and a green one here, of genes that were low on day 14 and gradually increased until day 23. So these are going up during the course of early pregnancy. And sure enough, all of these purple ones here are, its upstream regulator would be interferons. So pathways that increase represent interferon response genes, as well as some other metabolites. And the coolest thing was that pathways that decrease were pathways that we know are pathways activated by PGF2 alpha in CL. And so we think these genes in these clusters could be the genes that explain why the CL is less sensitive to prostaglandin in pregnancy. And also, these are not interferon regulated genes. So that tells us not all of the changes in the CL are due to interferon. There were also some that sort of went up and went down, and those had mostly to do with immune cell signaling, which I love because it means I was right. I wasn't wrong after all about immune cells, and it does exactly what we expect. That in early pregnancy, you have to have an immune response to that embryo because the embryo is part foreign, and you initiate an immune response. And if you don't have an immune response, you're not going to get pregnancy, but you have to program those cells. And in the CL, you have to have that immune response, but then you have to have it turned off right away. And that's exactly what we saw. Okay, and then I just wanted to show you one slide from the microRNA because I love microRNA so much. The microRNA that go up. Now remember, what microRNA do is inhibit proteins, right? The microRNA that go up are involved in PGF signaling. Those that increase over time are involved in PGF signaling pathways like calcium and phosphatidylinositol, all those things. So that means as those, during early pregnancy, those microRNA go up, and that's turning off or inhibiting translation to proteins that are necessary for PGF signaling. So again, that sensitivity of the CL to PGF is probably regulated by these microRNA. On the other hand, the microRNA that decreased are the ones that would target steroid hormone synthesis, which means you would have more of the proteins involved in steroid synthesis. So explaining why the CL of pregnancy has a higher storogenic capacity. So microRNA rule the world. But when you do all of this sequencing, one of the things that's really important is to think about what is the function of all these things, because you do sequencing and you run pathway analysis and you always get a new response and sell this and sell that, right, in everything you do. So one of the ones we chose were we had pathways that were involved in vitamin A synthesis. Now you saw what that CL looks like, right? That CL is that bright orange yellow color because it's full of beta carotene and like carrots. And beta carotene is converted to vitamin A, as you know, and so the CL can have a lot of vitamin A, but nobody had ever determined if the CL itself can convert beta carotene to vitamin A, and nobody's ever looked at vitamin A or retinoic acid signaling in CL. But we had indications from all of this transcriptomic work that we did that this might be the case. So this enzyme, ALDH1A1, converts retinaldehyde to retinoic acid, which is the one that signals, the active one. So you can see the ALDH1 protein goes up during early pregnancy, and then Camilla cultured luteal cells, and she found an ALDH1A1 inhibitor. And when you culture them with that inhibitor, so you inhibit this production of retinoic acid, progesterone production goes down. So from all those transcriptomic studies, we pulled out this one to do with retinoic acid and vitamin A, and we're able to show that it's important for progesterone production. Okay, so we're here, and we're going to end, I promise now, this argument. So Milo and I were still arguing, and we came back to this argument where he says that the uterus does not release prostaglandin F2 alpha in early pregnancy. And what I really wanted to do was do that experiment that Kipinski had done, where he put a cannula into the tail vein of the cow and ran it down to the vena cava, so you can measure prostaglandin coming, PGF2 alpha coming out of the uterus and the ovary. But you know, I'm old and I really didn't want to go out and figure out how to do all these surgeries because I know it takes years to learn a technique like that. So at this meeting, I ran into Kai Poehler, who's down at Texas A&M, and we're talking about this debate going on between me and Milo. And I said, you know, Kipinski did this stuff in cows with this surgery technique, and I would love to be able to do that, but I can't. And Kai looks at me and goes, I've been learning that surgery. He says, I would love to do this with you. So we started this collaboration, which is so fun near the end of your career to collaborate with some young, enthusiastic scientist with a lab full of students because they have done such a great job. So he runs a cannula into the tail vein of the cow and then ties it all up there. So here you can see this is what the cannula would look like coming in through the tail vein, gets it up here to the vena cava, which is where the uterine and ovary veins come in. And so he can collect blood samples from right there coming out of the uterus. So they've done this. We've been doing this off and on with different groups of cows for like 2.5 years now. And so they cannulate the cows on day 16. On day 17, they start sampling. They sample every 4 hours. And then for the next 2 days, we do hourly samples. And then every 4 hours out through day 20. And we have 2 periods where we did 20-minute samples. I'm not going to show you from that because I'm just going to show you a little bit at the preliminary data. So here we are. Here's all of his students out there who are my angels now for doing all this. Because, you know, they live at the barn for 4 days and he makes them do this between Christmas and New Year's half the time. But they sent us thousands and thousands and thousands of samples, and my technician has done thousands of ELISAs for prostaglandins. So I'm just going to show you 2 slides, and I want to tell you this is preliminary data. We've not summarized, we've not done any analysis of peaks and all that stuff, but here are 2 cyclic cows. And so all these slides, the top graph is the PGF2 alpha that was measured in the vena cava, and then the PGFN that was measured in the jugular, and then we also measured PGFN in the vena cava. We measured other stuff, but this is what I'll show. So here's a cyclic cow. And boom, there's her peak of PGF2 alpha. And you can see a coincident peak of PGFN in the jugular And then there's also some other PGFN in the jugular that we didn't pick up here. So that looks pretty good, except that if you remember that ester cycle thing I showed you, and we teach this, like if you look in Sanger's repro book, that PGF2 alpha increases in pulse amplitude and frequency, right? Don't we teach that? It's wrong. It does not. All the cows we have, it does not do that. You get one or you get two of these pulses. And so here's another cyclic cow. She had 2 pulses of PGF2 alpha. And then she had, again, a big pulse of PGFN, both in the jugular and in the vena cava that we didn't see. But, you know, those are cyclic cows and kind of what you would expect. Here's a couple of pregnant cows. Now, one of the things to note in the pregnant cows is there's a lot of noise. There's a lot of going up and down. But you can see here, this looks like there could be pulses here of PGF2 alpha coming out of the uterus in this pregnant cow, and maybe a coincident pulse of PGFN, but it's a little harder to see because in this one, the baseline is up here and there's a lot of up and down. But look at this cow. This cow had a pulse of PGF2 alpha from the uterus here and here. Big pulses here, almost 500 nanograms per mil, and 2 of those with really big pulses of PGFN in the jugular. So there's PGFM or PGF coming out of the uterus that was not really picked up in the jugular in this seed, and this cow remained pregnant. So they can have big PGF pulses coming out of the uterus and they can remain pregnant. So that's going to be our last great paper. So the CL of pregnancy is different from that of the cycle. Are those differences necessary for the CL to be maintained? You know, Milo says no, I say yes. So this goes on, this argument. But it could be that in addition to the uterine release of PGF2 alpha, there are other mechanisms that are required. So where do we go from here? And I'm going to end by saying that Milo called me up one day and he said, you know, you have these data of this time over pregnancy that you've done transcriptomics on CL. And we have data where we've compared CL with day 20 of the cycle of day 20 of pregnancy. What if we put our data sets together and collaborated and tried to come up with common genes that were differentially abundant, and those might be really involved in rescue of the CL? We even use that word rescue once. So we did that, and because of time, I'm not going to show you, but we did this. We found all these common genes. We found out, you know, they were the pathways that they were involved in were interferon signaling and retinoic acid activation came up in both of our data sets. And I just wanted to show that we took all of the genes that were involved in retinoic acid signaling from those pathways. We pulled out what those were. So in yellow is, and blue, and the 1 and 2 has to do with Penn State and Wisconsin data. And the bottom line is from that is, it wasn't always the same gene in the 2 data sets that was different, but when you put them together, you get a number of points of regulation of the retinoic acid pathway. So we're really convinced, we're all really convinced now, that retinoic acid that has really not been studied in CL is really important for luteal maintenance or rescue or whatever you want to call it. Okay, so what are the take-home messages? First of all, immune regulation is important for maintenance of the CL. Some effects of interferon tau on luteal cells are consistent with supportive steroidogenesis and survival. So we think interferon tau signaling directly in the ovary and the CL is probably important for rescue. The CL of pregnancy, we know from other data, has a greater capacity to make progesterone than the CL of the cycle. It's less sensitive to PGF and it synthesizes less PGF. Pathways that we know are activated by PGF are decreased during early pregnancy, and microRNA are very likely involved with these things. And now from these studies, we've discovered that retinoic acid is probably very important for supporting luteal function. During maternal recognition of pregnancy, in pregnant cows, PGF can be secreted from the uterus in substantial quantities, and pregnancy can be maintained. So that's why you're not allowed in your class anymore, Shelley, to say it inhibits prostaglandin. It modifies it, I think, but it doesn't inhibit it. And most of all, here's the thing that I really learned from all this. Scientific debate is healthy. So I see people go to meetings now, to science meetings, and they don't want, they'll go over to Savannah and say, you know, I don't need it, or I don't need it. But they won't go to Mary Alley and say, no, let's talk about your data because I think it could be a different interpretation. But it's really important because there's all kinds of great examples in our field of reproductive biology of scientific debate that the controversies have driven the most amazing science to get to the answer. One guy won the Nobel Prize, Ernie Nobile, because of this scientific controversy we got into with Harold Spies. Ernie Nobile and Harold Spies stopped speaking to one another. They wouldn't be in the same room at the same time. They went to their graves as enemies, right? So it can be really, really good, but you don't want it to go that far because the last one is Milo and Joy are still friends. So we're not going to go to our graves as enemies. We talk, we've enjoyed this controversy. We've tried to actually collaborate to work it out, and we continue with that. So I just will end. I'm sorry I went a few minutes late here with thanking. I want to thank many grad students who have contributed all along the way, but the data I show you today mostly are what Camilla Hughes had done in these last studies. Keith Inskeep, who helped us with that experiment I showed you, and we couldn't have done that without him. My technician, Casey Baker, who is so sick of running prostaglandin assays now, but we're getting to the end. We're almost done those. Certainly Kai Polar's lab. But you know, most of all, I thank Milo, because Milo challenged my thinking. He made me think different. And that study that Camilla did of the temporal changes over pregnancy, we had all of those data. We hadn't published them. We were thinking through, we were analyzing, doing the data interpretation. After I sat and argued with Milo, we totally changed sort of the approach we took in that paper, and the paper is so much better because of it. So I really thank Milo because he challenged my thinking, he made me think differently, and he energized me to do a different kind of science and find a collaborator and actually work on things that come out of the uterus for a change. So with that, I'll stop and hope you got a few life lessons out of that.
All right, thank you. We're going to take time for questions, but I realize some of the students might need to leave. Feel free to do that. I'll turn the lights on so you don't trip on the way out. But, anyone want to start us off with questions?
Thank you for the some normal life lessons.
You're welcome.
But I was thinking as you were showing the graphs, the PGF alpha and the PGFM. So is there any value in seeing your data now and then?
Yeah, it's a good question. And this is, so I presented this in Italy and then I presented it in Utah Biennial Repro Symposium, and Pete Hansen sat back there and said, oh my gosh, so does PGF not, kill the cell or something? It's like, no. Is interferon tau not important? Yes, it's important. So I know that we can get PGF2 alpha, and here's what I think happens. Interferon tau is modifying prostaglandin release. Now you will note I didn't show any summarized data there, and all of the y-axes were different, right? And that's because we have to run these ELISA plates. We have to run one cow all for samples sequentially to catch the peaks. And there's huge plate-to-plate variation in these ELISAs. They're terrible that way. So what we decided to do is we're running through all of them. And then at the end, we're going to choose samples from all of the cows and run them together in one plate so that we can then compare how much PGF is in the pregnant cows and how much is in the cycle cows. But what I do know is it's hugely variable. And so I think there's not going to be perhaps that much difference. But in the pregnant cows, I think interferon tau is decreasing prostaglandin somewhat, which is helping that CL to not regress. But pregnant cows can make prostaglandin, which is why we need these other mechanisms to make that CL less responsive. So I think that CL becoming less sensitive to prostaglandin is another thing that happens along with the decrease or the alteration of prostaglandin release from the uterus. And when you think about it, I mean, what is more important to survival of a species than pregnancy, right? You have to have pregnancy. So why would ruminants have evolved so that there's one thing that gets altered to ensure pregnancy? It just doesn't make sense to me. And so we know about prostaglandin and it's changed, but it's not all gone. I am so convinced of that. You know, in our pregnant cows, we have pulses of PGF, but those cows maintain their pregnancy, so that CO is not responding.
Did you have a question?
Yeah, I was curious about this difference in the luteal cell size. Are these like subpopulations that stay big or small? And are they like changing? Are there any functional differences to being a small luteal cell versus a
Yes. Yeah, so that was a huge topic of investigation when I was in grad school. There were two labs, one at Cornell, one at Colorado, one did cows and one did sheep, and they were studying small and large cells. They were separating them and finding out what's different about them. This was another big controversy where they were trying to do each other. So basically, it starts out, those cells that were in the follicle, there's two kinds. There's granulosa cells inside the follicle, right outside the basement membrane. The granulosa cells become the large luteal cells, and the theca cells become the small ones. But there's also evidence that the small ones over time grow up to be big ones. And they are functionally different. They both make progesterone. The small ones respond much more to LH from the pituitary to drive progesterone. The large ones are more constitutively active, so they make high amounts of baseline progesterone. The large ones respond better to PGF2 alpha. The small ones don't have very many PGF2 alpha receptors. So we know there's a lot of like crosstalk between these two cells. So yeah, it's good thinking because all of those questions you had, yes,
biologist, I'm a cell biologist who studies cancer cells that are different size.
Yeah, but you were right in every question you answered. Yeah, as you went through that, I thought, yeah, the answer is yes for all of that. They are functionally different. Yeah.
Thank you.
About the prostaglandin story, I think Keith Inskeep could be claimed for this, that he had a big discussion point about PGE changing in early pregnancy as well. Have you thought about including PGE into all this stuff you're doing with Kai?
Yeah, so we've measured it.
Okay.
So we're running PGE from Vena Cava as well.
Okay.
Yeah. So far, it hasn't looked as interesting. Like I haven't seen any big differences between cyclic and pregnant ones. But what we thought based on Pete's data is that in the pregnant animals, we would get more PGE. We're measuring PGE2. So that you would get more PGE2 or a higher ratio of PGE2 to PGF2 alpha. But until I can sort of normalize across all of these animals in individual plates, I can't really make a conclusion. So we have the data.
Interesting.
Yeah.
Bill Beal, retired faculty member in animal science here. It was great. It's like deja vu all over again. The question I have is totally different from these others, and that is as you look back on the graduate students that you have trained, what characteristics are most important for success of a graduate student? Number one, and then what are the, like when someone asks you about graduate school, what do you ask them about in terms of their expectations after graduate school? So those two things, characteristics, and what do you ask them about their expectations?
Yeah. So thank you for those questions. I spent yesterday, I was so thrilled that Alan set me up to talk to grad students. I said, it's fun talking to grad students because they're happy and faculty are not happy. So it was a very fun day. So I've been thinking about this. So let's see. The characteristics of grad students that I think make them really successful. So first of all, I'm going to divide this into master's students and PhD students. And I'm a big proponent these days of master's degrees, which many of you have heard me talk about already. So won't go into all the reasons for that, but I think I look for different things in those. So for a master's student, I'm looking for a desire to learn and a desire to start to understand how things work and to be really good in the lab. So a master's degree in my lab is really learning how to do things in the lab and become proficient and validate what you do and learn about your data. And then master's students may or may not choose to do a PhD. And so that's when I start asking, I'm going to come back to the first question, but the second one What is it you want? Because I fear that we are driving too many students into PhD programs who don't need to go into PhD programs to get to their final goal. And the last thing you want is a student who's in there just to get a degree, and it's not a degree that they need. So I tell them there's a lot more jobs out there for people with master's degrees, research lab-based masters in agriculture-based companies and in bioscience companies, tons of jobs, good paying jobs. For a PhD student, I think the biggest thing that I think makes them successful is if they have a passion for science. And along with that is a curiosity. Because you can have a straight A student, a very bright student who can learn all of these things that you teach. But if they don't have that passion that is required to really delve in and start to think about your data and think of it in light of everything that's else that's known and know what the next questions are, be able to want to figure out what those next questions are to be asked. Those are the students who do that, who really succeed, who really shine. Camille Hughes was one of those students. The students who don't do that, who don't ask the questions, they're not happy. And I've had these, they're in there because they think they have to get this PhD, but they're not happy in grad school and they're not going to shine. They're not going to do really well. So I think passion and curiosity are the most important. Yeah.
There's somebody online with a question, Pedro Fontes. Pedro, I think I gave you access to ask the question. So if you want to unmute yourself and try, we'll see if we can hear you.
Sure. Sure, guys.
Yes, we can.
All right. Well, thanks, Dr. Pate. Very, very nice talk. I had a quick question. So it seems like in your studies, when you're looking, the one you share later with Kai Poehler, that you're profiling prostaglandin in cows that became pregnant versus cows that did not become pregnant. So where do you think pregnancy loss fits in all that from a cell biology standpoint? We have some cows that they might undergo, conceptus elongation, potentially even have onset of placentation. We can maybe pick up an increase in PEGs in circulation around day 20 of gestation or so. So where do you think all that kind of meets together? The whole pregnancy loss idea relating to your cell biology type of work, at least at that part of this gestation.
Yeah, thanks, Pedro. That's a really good question. I don't know the answer. What I think is that because I believe that you have these multiple mechanisms in play in pregnancy, that if any just you don't get the right balance, that you might lose that pregnancy, whether it's alteration of prostaglandin or sensitivity of the CL. But what I can tell you is we have 2 animals that we collected samples from that were bred but ended up not maintaining their pregnancies. So we call them bred non-pregnant. We haven't started to analyze those samples. We're saving them to the end, and there's only 2 animals, but there might be something telling in that. If there are things that we can find that we can categorize as these hormones that occur in cyclic cows and then what occurs in pregnant cows, we'll be able to see in the bred non-pregnants, maybe if they fall in terms of those hormones more with the cyclic or with the pregnant. So maybe those 2 cows will tell us something. Otherwise, I think Kai's going to have to go back and collect more samples and collect the ones where the pregnancies fail. But it's a great question.
Yes, thank you. Thank you.
Okay, are there any other questions?
Thank you, Dr. Pate. The PGM that you measure in the blood, is it only metabolite for prostaglandin F2 alpha, or is it metabolite for other kinds of prostaglandin as well?
Okay, so I'm smiling because you're smart. So for prostaglandin F2 alpha, it gets converted to PGFM, which is 13-14 dihydro PGF alpha. But what you're going to say is we call that the stable metabolite of prostaglandin, and that's what everybody measures. It actually, PGFM can be further metabolized to these things called tetramer prostaglandins that nobody measures. The other prostaglandins have their own metabolites that are similar. So for like PGE, there's a PGEM. But the PGFM we measure is only the metabolite of PGF. Now, I will tell you, all of us, which is not very many, who have run various PGFM assays, and every one of them out there sucks. But none of us think that the PGFM antibodies are good. So if somebody wants to make a PGFM antibody that's any good, we'll get much cleaner data.
Because it would kind of explain your weird pattern of PGFM not in relationship to the peak of PGF that you get.
Right. So it could be that we have some cross-reactivity of something. And so I tell you, you have to validate everything. We're using Cadence kits. And so you get cross-reactivity info. There could be something maybe that cross-reacts there. It also could be that if you don't have a coincident peak where we're doing hourly samples, we just missed it a little bit. That could be. And it also could be when we get peaks of PGFN that we didn't see a peak of PGF2 alpha and beta theta. It's because prostaglandins are made all over the body and they're metabolized in PGFM, and PGFM can come from all sorts of places. So it could be that we're just something else happens in that cow that we pick up PGFM.
Any last questions? Last chance. Well, thank you very much.
Thank you all.